Thanks, Roseanne.
Are we good?
Yeah, so if you're okay, I'll just introduce you very briefly, as I mentioned to you.
Yes, please go ahead.
So welcome everyone for today's talk. That's part of the VT Life Science Seminar Series that today will be presented by Dr. Harris Lewin. I did mention to him that I'll do a very short presentation of his accomplishments, which will not do justice to how accomplished he is. So I'll try to make it short. So Dr. Lewin spent 27 years at the University of Illinois at Urbana-Champaign, where he held an appointment at the Department of Animal Sciences with a prominent endowed professorship in immunogenetics. Dr. Lewin, also at UIC, served as a director of the University of Illinois Biotechnology Center. He was the founding director of the CAG Center for Comparative and Functional Genomics. And more importantly, he was the founding director of the really world-renowned Institute for Genomic Biology. He unfortunately or fortunately left UIUC, and in 2011, he joined UC Davis as a vice chancellor for research that he served until 2016. Thereafter, until 2022, he was the Robert and Roosevelt Osborne Endowed Chair and Distinguished Professor of Evolution and Ecology at the University of California. And now, more recently, Dr. Lewin is a Distinguished Professor Emeritus of Evolution and Ecology at the University of California, Davis. I'll just touch on some of his prominent honors. In 2008, Dr. Lewin was elected as a foreign member of the Royal Swedish Academy of Agriculture and Forestry. In 2011, he was awarded the Wolf Prize in Agriculture. And in 2013, he was elected to the United States National Academy of Sciences. More recently this year, Dr. Lewin received the Lowell Thomas Award in Conservation Genomics from the Explorers Club of New York. And more specifically related to today's talk, in 2017, Dr. Lewin co-founded the Earth Biogenome Project, which we'll hear from him the latest today. And he currently serves as a chair of the Earth Biogenome Project Working Group. So, Dr. Lewin, I cannot tell you how appreciative I am for you to spend this hour with us. And then I'll let you start your presentation. Thank you so much.
Sure, thank you. Can you hear me?
Yes.
Great. Yeah, so what Fernando left out was, of course, that he was a postdoc in the lab for, for 4 years or so, and it was just wonderful to have Fernando there. And in Fernando's honor today, I'm going to take off my sweater and show you that I'm wearing my Brazil colors for the game, the World Cup today. And there's also some selfish motivation there, Fernando, because as Fernando knows, my wife is Brazilian. And so this is a matter of survival for me to wear the colors. But tomorrow, I'll be wearing my colors for the game. Here we go. See, Fernando, we're going to be rooting for the USA tomorrow. So anyway, I can't hear if you're laughing in the crowd or crying, but today we're all wearing the yellow for Brazil. Okay. So thanks again for your introduction, Fernando. And as Fernando mentioned, today my talk is going to focus on primarily on the Earth Biogenome Project in the first part. And I'm going to talk just a little bit toward the end about how the reference quality chromosome scale assemblies that we've been generating in the Earth Biogenome Project have been used, particularly in my lab, to study the problem of chromosome evolution. So I'm not going to talk about, more than 25 years of research on cattle genomics and many of the exciting discoveries and applications that were made along the way. But ultimately, that work that we began at the University of Illinois more than 25 years ago, 30 years ago, is what led to my current interest in genome evolution, which has been really my passion over the last 20 years. And I'm very happy to talk about that with all of you today. And so it is now generally accepted that biodiversity is facing an existential crisis, which has been brought about by changes in land use, pollution, overexploitation, habitat destruction, invasive species, and now what is turning out to be the greatest threat of all, which is climate change. The International Union for the Conservation of Nature has surveyed more than 147,500 plant and animal species and has determined that 41,000 species, or about 28% of all the assessed species, have undergone dramatic reductions in population size and therefore under a direct threat of extinction. And science has shown that the rates now of extinction of species are 100 to 1,000 times above the background rate that we can determine from the geological record. And if things keep going the way they're going, it's predicted that by the end of the century that we may lose up to 1 million species of plants and animals. And if that happens, most of the ecosystems on which we depend on the planet could be under severe threat, and a loss of 1 million species due to climate change would likely be a tipping point that would lead to ecosystem collapse on a global scale. And that would threaten the existing world order and probably even our own survival as a species. So why, again, yes, so the predictions of biodiversity loss now have, and the recognition of the link between climate change and biodiversity loss has brought about unprecedented activity on a global scale to preserve biodiversity. In fact, this week, as many of you know, in Montreal, there's going to be a meeting, the 15th meeting of the UN Convention on Biological Diversity. And this, what's called the COP15 meeting, will focus on setting agreed global targets to halt and reverse the loss of biodiversity. So this is becoming an increasingly important challenge. And the reasons for it is that biodiversity provides the basis for ecosystem services that support life on Earth. Ecosystem services that support our agriculture, clean water, and all of the regulation of Earth's biogeochemical cycles depend on biodiversity. And biodiversity is also the foundation through agriculture and the food industry, the biochemical industry. It's a foundation for the global bioeconomy. In the United States and in the European Union alone, more than $500 billion per year, estimated in the year 2017, is of the global bioeconomy is dependent on biodiversity. And if you look at probably the total annual economic impact of biodiversity, it's more in the range probably of $3 to $5 trillion globally each year. So protecting biodiversity is really now a priority for the entire world. And if you think that protecting biodiversity is important and biodiversity is important, you would think that understanding of life on Earth is also very important. But our understanding of life on Earth is severely constrained. There's an estimated 7 to 10 million eukaryotic species on Earth, of which only about 20% even have names. And of the possibly 4 million, probably this is a tremendous underestimate of bacteria and archaea, less than 1% have been identified. So even though we have such high dependency and biodiversity is so important for our existence and well-being, we actually know very little about life on Earth. And a lot of what we've learned especially in the past 3 decades, about life and how life works and the total interconnectedness of all life on the planet has been due to the revolution that has occurred in genomics, again, in the past 30 years or so, that has allowed genomes to be sequenced and genomes to be annotated for their function. And much of this has occurred through comparative genomics, through comparing the genomes of living organisms from bacteria, for example, to humans. And the revolution in genomics has been brought about by a rapid development, as you all know, in genome sequencing technology. Sequencing technology does progress to lower the cost over 1 million-fold over the last 30 years or so. So the human genome cost about $3 billion to sequence, and today you can sequence a genome of better quality than you could that was published in 2003 as the quote draft, a good draft of the human genome. You can do that for about $1,000 today or less. And so the sobering fact is, even though we've made all this tremendous progress in genome sequencing over the last 30 years, less than 1%, but 0.7% of known eukaryotic species have had their genome sequenced. And of those, less than 10% are of the type of quality that we have become accustomed to when we study the genomes of agriculturally important species of plants and animals, less than 10%. So we really, of the 0.7%, so less than 0.1% have reference genomes. So this is a tremendous constraint, and we're missing the critical information that is necessary, the fundamental knowledge that's necessary to understand biodiversity, to reduce and reverse species extinctions and stabilize ecosystems, and the fundamental information that is necessary to develop the strategies and science-based policies for biodiversity conservation. Now, in response to that, and in response to limitations that many of us were facing in our own research as a result of not having high-quality genome sequences to compare, we proposed back in 2018, actually the work for this began in 2015. It took about 3 years to develop this plan, which we called in 2018 the Earth Biogenome Project. The manifesto was published in PNAS in 2018, and the project was officially launched in London later that year at the Wellcome Trust. So what exactly is the Earth Biogenome Project? The Earth Biogenome Project is a confederated international network of networks that has the common goal of sequencing and annotating the genomes of all named 1.8 million named species in 10 years. So it's bounded by a time frame, so it's presented as a major scientific grand challenge with the goal of creating a complete digital repository of eukaryotic life on Earth, representing the collective biological intelligence of 3.5 billion years of Earth's evolutionary history. And we've promoted the view that this knowledge will guide future discoveries for generations. It will provide the foundations, the basic infrastructure for the future of biology and a future sustainable global bioeconomy. And the project, when it was announced in 2018, generated a lot of interest in the popular press and in the scientific media. And since that time, we have been busy at work in the pilot phases of the project. And I can steer you all, if you're interested, to a series of 10 wonderful papers that were published in a special feature of the Proceedings of the National Academy of Sciences just this January, outlining the progress of the first 2 years of the Earth Biogenome Project. And so I won't go through all these papers, but of course there's a summary paper, and there's also a very nice paper which describes the scientific reasoning and rationale for sequencing them all rather than sequencing one particular branch, say the animal, the metazoans, or plants or fungi, or the protists. So we are dedicated all to sequencing all of them. Since that time, the Earth Biogenome Project, this confederated international network of networks, has grown to include 43 member institutions, a total of 51 affiliated projects. Most of these projects deal with, are specializing in particular eukaryotic taxa. These 51 projects are located in 22 different countries, have more than 5,000 participants, and the participants themselves are located in more than 110 countries. So the projects are based in 22 countries, but the participants are now in more than half of the countries of the world. And the project is again organized as a global hub and spokes model. The members of the project are committed to open data access by signing the Memorandum of Understanding. They're committing to open data access in compliance with the Convention on Biological Diversity, what I mentioned at the start, because you just can't go in and anymore and take samples, even for purely for scientific use. You have to collect according to certain protocols, and there are reporting schemes, and it has become quite difficult to do collections around the world. And so we're all committed to open access and compliance with the CBD, and committed to diversity, equity, inclusion, and justice among the EBP communities and scientists and peoples of the world. As I mentioned, it's a hub and spokes model. So we have sequencing nodes that are located in major biogeographical regions of the world, taxon-specific affiliated projects, and all of that is coordinated by a central coordinating committee. I'm very fortunate to chair this international committee, and one of the central roles of the committee are to develop standards for the project, for sample collection, for sample processing, for sequencing and assembly, and annotation and downstream analysis. And this secretariat for the project is currently located at the University of California, Davis. And this was not, of course, done alone. I just want to take a minute to acknowledge the many people who have been involved in the leadership of this project. First, my co-chairs for the project, John Kress, who was former Undersecretary of Science, a plant ecologist at the Smithsonian, and Gene Robinson, an entomologist, member of the National Academy of Sciences, and chair and also the other founding co-chair of the project. We have several major committees that are responsible for the governance of the Earth Biogenome Project. We have an Ethics, Legal, Social Issues Committee that's chaired by Hank Greeley at Stanford and co-chaired by Melissa Goldstein at George Washington University. Federica De Palma, Genome Canada, British Columbia, and the Earlham Institute chairs our International Scientific Committee. And Sadie Paez and Marcelo Ileana da Silva are co-chairs of our Justice, Equity, Diversity, and Inclusion Committee. Sadie's at Rockefeller University, Marcella's at the Sanger Institute, and Nicolette Caparello at UC Davis here, who is our executive manager and chairs the Communications Committee. Now, the job of developing the standards falls under Federica's committee, the International Science Committee. Mara Lonachak at Sanger, it chairs the group on sample collection and processing. Richard Durbin, very well-known mathematician and bioinformatician, is the chair of the sequencing and assembly group. Francois and Fergal co-chair our annotation group. Francois at NCBI. Many of you may know her if you've tried to submit annotated genome sequences, and Fergal's at the European Bioinformatics Institute. Justin Lindblad-Toh at the Broad Institute and the University of Uppsala as chair of our data analysis group, and Xiaofeng Wei at BGI in China is the chair of the IT and informatics group. And this is basically forms the leadership of the current leadership of the Earth Biogenome Project. Now, to remind you, for those of you who haven't been following this, the project doesn't propose to just sequence all 1.8 million species within 10 years, but we have phased this in a phylogenomic wave. We actually have two strategies. I will only talk about the phylogenomic wave today. Phase one is the sequencing of a representative member of each eukaryotic taxonomic family in the first 3 years. There are about 9,600. These are the numbers now in NCBI taxonomy in parentheses here. There are other taxonomic databases, but we're currently pretty much hooked on NCBI's taxonomic classifications. There are 9,600 eukaryotic families, and those will go in the first 3 years, will be sequenced in the first 3 years. Phase two in years 4 through 7 will involve sequencing of the 120,000 representative species of each of the 120,000 genera. And then in the out years, the sequencing of the remaining 1.58 million. And should we get to that, of course. And this project is not going to be inexpensive. But when you look at the cost of the project, which has been through rigorous, actually, testing and analysis, we initially estimated in 2018 a total cost for this project of $4.7 billion over 10 years over the entire world. And the original, if we look at the cost of the human genome sequencing of the human genome in today's dollars and not the 1990 dollars, today's costs are approximately almost $6 billion. And so the cost of sequencing all of eukaryotic life today, and that's all in costs, including computation and reagents and collection of the samples, is less than the cost of the sequencing of the human genome. And with a return on investment, which was estimated at 65 to 1 in this famous Battelle report of 2013, probably an overestimate, but at least 30 to 1 today, just for the human genome. I mean, imagine what the return on investment is going to be over time for sequencing all of eukaryotic life. In fact, I would argue that the Earth Biogenome Project is probably the greatest bargain in the history of science. And costs keep coming down. And even today, it could cost, for some samples, it could cost more to collect the sample than it costs even to sequence it. So this is rapidly changing. And changing, the numbers are changing in our favor. In fact, we can do higher quality sequencing at lower cost than we'd originally anticipated, and perhaps be able to develop a reference quality genome for all eukaryotic species. Okay, so where are we with the with the Earth Biogenome Project? What has the progress been during the pilot phase of the last couple of years? Well, I'm very happy to tell you that we have now surpassed in the pilot phase over 1,000 genomes. So we're at 1149 as of last week. And these 1149 species represent 547 unique eukaryotic families. And that represents about 5.5% of the total eukaryotic families. So the pilot phase has done pretty well in our goals. Yes, we have twice as many species as we have families, but that is rapidly changing as we begin to focus most of the projects toward those phase one goals, toward the phase one goal of producing one representative species for each taxonomic family. And as we move to the additional phases, we are going to face many, many challenges. I'm going to just speak briefly about those toward the end. But right now, the first phases are moving along very rapidly. And here's some additional data to show that. In fact, these are the numbers over the last few years just for the Earth Biogenome projects, the 25 projects that I mentioned previously. And I might add one other fact here is that we are grossly underestimating actually the number of species that are sequenced by the EBP affiliated projects, because we only have half of the projects that are integrated into the goat genome database. Goat is genomes on a tree, and this database has been created by the Wellcome Sanger Institute in the UK, and is allows us to track progress across all of the 25 projects. And we're only track all of the projects, and we're only tracking about 25 of the projects so far. So this is an underestimate. I think we probably have about 2,000 species actually sequenced, closer to 1,000 families if all the fungi of the DOE's JGI fungal genomes project are included. But as you can see, the number of genomes chromosome scale assemblies has been going up rapidly at the expense really of just the scaffolded and contig-based assemblies here. So here is where the standards are kicking in, and the cost of long reads and high C have been coming way down. And so we're doubling in the past few years the number of species and families, and the output is about across the project about 1.5 genomes per day. And actually that global capacity now is rapidly increased. We're seeing numbers that are approaching 5 and 6 genomes a day. By this year, we expect to be at about 9 genomes per day. And that's what we're going to need, 9 genomes per day, every day, 365 days a year globally to achieve the phase 1 goal of 9,600 representative species for 9,600 families in a period of 3 years. Okay, and what I think we're really proud of as a project is that if you look at the numbers of reference of genomes that are meeting the minimum reference quality standards that the project has set, and that is a contig N50 of at least 1 million bases and a scaffold N50 of at least 10 million bases. The Earth Biogenome Project versus the rest of the world has produced about 42% of all of genomes meeting this particular quality standard, which is a basic quality standard for reference quality genomes. And if you look over at families, this is the number that is rapidly increasing in our favor. We're well over half, approaching 60% of all of the reference quality genomes at the taxonomic family level have been contributed by EBP affiliated projects. And again, that is in fact an underestimate, because not all EBP affiliated projects have been integrated into the GOAT system. But this is all great news. And you might ask, well, who's doing all these genomes? Well, the leading lab in the world is the Wellcome Sanger Institute at the moment. They were the first to get funding for an Earth Biogenome Project related project, which they call DTAL, or for Darwin Tree of Life. And as of last month, they have collected fresh collections of 6,100 species. You just can't go to a museum and take dried specimens and get reference quality genomes. You really need to have very high quality samples to get high quality, high molecular weight DNA that's necessary for the long reads that produce these highly contiguous chromosome scale assemblies. They just last week had a party out at the Sanger in which they celebrated going over 500 species in really just in the past year and a half, representing 156 families. Most of these are Lepidoptera. They've done over 120 families of Lepidoptera so far, and 3,000 species are in progress and should be completed in 2023. And as if you can see, this is that quality metric zone of N50 1 megabase contig versus N50 scaffold of 10 million bases. Nearly all of their assemblies are falling in within the EBP the quality metric set for EBP for reference genomes. And so hats off to my colleague, Mark Blackster, who's the director of the Tree of Life program at the Wellcome Sanger Institute, and his entire team. Now, another major contributor is the Vertebrate Genomes Project. Most of these projects are linked in one way. VGP collaborates with Sanger, and Sanger collaborates with VGP. I'm a member of this because I'm interested in mammals and other vertebrates. And this is a project that's led by Eric Jarvis at the Rockefeller University. And so far, if you go, you will find 106 vertebrates species reference quality genomes submitted in INSDC databases. That would be GenBank, for example, or the European, what is it, the EBI database. I'm just blanking on the name of it for a minute. But in fact, there's over 200 that have been submitted. There have been about 106 released. So Eric and his group at Rockefeller are really making rapid progress toward sequencing one representative of each of the vertebrate orders. So that would include orders within the fishes, the mammals, the reptiles, the birds, and what did I leave out? And amphibians. So the target of the VGP is to produce a reference quality genome for all of the approximately 60,000 species of vertebrates. Very well organized. The reference quality genomes are really coming now. Again, this is the EBP metric, and so things are looking really good. And these two labs have been the major producers to date, but as I mentioned, there are 25 projects. We have one out here in California that's producing 150 reference genomes for conservation. There's a new project in Europe, Biogenomics Europe, that will be producing about 500 reference quality genomes across the European continent. Australia has 5 projects producing several hundred reference quality genomes, mostly for conservation, and so on. Norway has a national project, Sweden has a national project, and things are going very well. And we hope to have national project here in the US too within the next year or so. Now, so that's I'm about halfway through here, and I want to tell you about some of the recent work that we've been doing in my lab using chromosome scale and reference quality genomes to answer an important biological question. And so this second part of my talk will focus on use of reference quality genomes to study a very fundamental problem in biology. I'm going to talk about the work of my most recent postdocs, Joanna Damas, and a large group of collaborators who contributed genomes to this project through the Zoonomia Consortium led by Eleanor Carlson and Justin Lindblad-Toh. And we're going to see, I hope, convince you of the tremendous utility of these reference quality genomes for evolutionary analysis. So, as I mentioned at the beginning of the talk, I am interested in chromosome evolution. And I define chromosome evolution, for those of you who are not aficionados of this, as the process by which chromosomes change in the order and the orientation of their genes, regulatory elements, and three-dimensional organization and conformation over time. And we actually began studying this problem more than 20 years ago. And so my interest in this goes back to the early days when we first created our cattle maps that were eventually used to assemble the first cattle genome sequence. And when we were involved in the early days of comparative genomics, there were very few reference quality genomes to compare to. We only had, at the time, this was around the turn of the millennium, 2003, we had only the human genome in terms of mammals. We had only the human genome, the rat genome, and the mouse genome. And we had to compare them. We had to learn how to utilize synthetic information from maps from the other species that were available. So in 2003, when we began this work of large-scale comparison of mammalian genomes, there were really only 8 genomes that could be compared, and several of those genomes, like the horse genome and a few other of the mammals, were really of very poor quality and did not allow us to compare huge sections of the horse genome to other genomes, for example. And we were able to develop the tools and technologies to do that, including these visualization tool here, which you see, which we call Evolution Highway. And we could begin to deduce ancestral segments that would be common to all of the species that we compared. And those different ancestral segments are demonstrated in this particular cartoon by these colored blocks overlaid onto an ideogram of, say, human chromosome 1, human chromosome 2, and human chromosome 3. A rather odd way to view evolution of genomes, but these were the tools that we had at the time. And these comparisons led to actually many important insights, and we followed up on these studies and compared more genomes, up to maybe a dozen genomes over the next few years, got included in this analysis, but we quickly hit a wall, right? We hit a wall because there was simply not enough genomes to compare. And that was a big problem for us, and was a big problem for us for many years. And that was basically because of the cost. When we, at about 2005, or the costs were maybe $150 million to do, $100 million to do a genome. Those costs went down rapidly with some new advances in technology. Some of you may remember 454 technology. And, we were able to do genomes at the end of that decade for about $150,000. But we were really stuck for a while. And I'm going to skip really 15 years and tell you what we have been able to achieve in the last few years with this rapid onset of new technologies. So in that previous paper, we were able to actually get to the Boreoeutherian ancestor. In this most recent work, and the interest over the past 20 years is to reconstruct the ancestral karyotype and synteny groups on a sequence-based level for the mammalian ancestor, the common ancestor of all extant mammals that lived approximately 180 million years ago. And this is no trivial task. What we did was we had to have a reference or near reference quality genome for a representative species of each of the mammalian orders. And what you see here is that we were able to obtain or create ourselves reference quality genomes for all of the eutherians, all 19 of the eutherian orders, 3 of the 7 metatherian orders, and the platypus as our representative of the monotremes. Of the 32 genome assemblies that went into these reconstructions, 13 were chromosome level assemblies, 11 were chromosome scale scaffold assemblies, and 8 were scaffold assemblies, and 2 outgroups were used for these reconstructions. Now, for the first time, we created 3 independent constructions and compared them. There's only time to show you one of them today, but we also used the sloth and the cattle. These are 3 different lineages, which were very important to compare and validate the reconstructions. So, of course, the humans are in the Urocanogliers, the sloths are in the Xenarthran group, and cattle are in the Laurasiatherian superorder. And to do this, we use the genome assemblies, compare them using tools, some very standard and some very specialized. The most specialized tool is the descrambler algorithm created by my colleagues, Jae-Boom Kim, when he was a postdoc in the lab at the University of Illinois, now a faculty member in Seoul in South Korea, and Jian Ma, who was a colleague at the University of Illinois, now at Carnegie Mellon Institute. And this descrambler algorithm, basically what you do is you find all the similarities between a target genome and a reference genome, in this case, say the human genome. You find all the similarities, you do an alignment between every one of those other genomes and the human genome, all the genomes to the sloth genome, all the genomes to the cattle genome. And then the descrambler algorithm is a probabilistic algorithm, basically looks at all of the adjacencies of the synthetic blocks, and it uses phylogenetic information and outgroup information to determine the most likely order of these synthetic blocks in a common ancestor. And so we're able to do this for 15 different nodes on the tree leading to the extant species of mammals and this common ancestor of all mammals that lived 188 million years ago. And this is basically a picture of 188 million years of chromosome evolution in a single picture. And I know this is impossible to see. The details are not that important unless you're interested in particular chromosome rearrangements. But what you can see is the reconstructed mammalian ancestor, which had 19 autosomes and likely had X and Y. And then the colors represent correspondence between the mammalian reconstruction and the next reconstruction in the phylogenetic tree, which was the Therian ancestor. And if you see like crossing of the lines, those represent inversions. If you don't see any connections between blocks on different lines here in the different ancestor, that means that the entire synteny block, the entire group, was conserved. If you see parts of chromosomes going from one to another, those are either fissions or fusions or translocations. And this was done across all 188 million years. And in this case, I don't know, approximately 8 or 9 or 10 different ancestors. And the data are summarized here in this table. So if you want to try and understand what happened between the mammalian and the Therian ancestor, you would have a branch length here of 18 million years. There were 90 inversions, 3 fissions, 3 fusions for a total of 96 rearrangements. And that could be done at each step here. Each of the total rearrangements can be summarized, and we can compare rates of rearrangements across the different lineages, normalized for a million for each million years of evolutionary history, and then rates of evolutionary change could be determined across those phylogenetic trees, and we can find periods of geological time where rates were significantly higher or lower. And so this has all the rearrangements from the mammalian ancestor to human. So this is along the human lineage, and we were able to calculate 310 inversions, 30 fissions, 25 fusions, for a total of 365 rearrangements over 188 million years. And for us, that was very satisfying. It was a major achievement. And but there's lots that we did with this information. One thing which we learned was that we were able to identify, using these 3 independent reconstructions, that there were about 2,500 synthetic segments. These were multi-species homologous synteny blocks. These were blocks of genes that are on average of nearly 1 million bases that were conserved in all of the 32 genomes that we studied. And what these 2,500 blocks represent are the building blocks then of all extant mammalian genomes. And what's happened over evolutionary time is that these segments have reshuffled themselves in the different lineages. Sometimes you have tremendous degree of conservation. We found many segments that were over 10 million bases, some over 20 million bases that could go back 188 million years, and some that were even chromosome scale. We found 9 large multi-species homologous synteny blocks that corresponded to avian microchromosomes. Okay, so those synthetic blocks could go back over 300 million years. So those are blocks where the genes have not changed in their order. So you might ask, one question you would ask is, why might that be the case? And I'll only have really time to show you one of the many figures in that paper, but I think this tells you something about chromosome evolution that's not just apparent from the sort of this jigsaw puzzle of blocks that are shifting and moving over time. So you can do a geo-enrichment analysis, something that Fernando knows a lot about, for enrichment in multi-species homologous synteny blocks, these blocks, versus the breakpoint regions between the blocks. And one of the things that we observed, and here are the, these are very highly significant results. These are FDR corrected p-values. And one of the very clear observations was that the multi-species homologous synteny blocks, these are blocks that have not changed over hundreds of, in gene order, over hundreds of millions of years of evolutionary time, that these are related to anatomical and central nervous system development, regulation of developmental processes, cellular developmental processes, cell differentiation, neurogenesis, whereas the analysis of the genes within the evolutionary breakpoint region shows a very different picture. This is where the transcription factors are, and the genes related to adaptation, and genes that have been related to species-specific differences. So these breakpoint regions between the blocks are cauldrons of evolutionary change in the genomes, whereas these blocks, the order of the genes are maintained very stably over time. And we have proposed that this is due to one of two things, one of two hypotheses. One is that these segments may have lack the signals. You're unable to break the segments, or you're able to break the segments with the many meioses that would occur in a species over millions of years of evolution, but that those breakages would be subject to purifying selection. And we think that the latter hypothesis is the correct hypothesis, because if you are going to break regions that have highly conserved functions in animal development, this may then not be compatible with survival of a particular organism and would be subject to purifying selection. So it's just a nice example of one of the many things that we've learned by doing chromosome evolution studies. And I'm just going to say, we started this out, it was simply an exercise, a real intellectual curiosity. I mean, no apparent practical significance for studying why, chromosomes have evolved the way they do. But in recent years, as many of you have probably been following in the popular press and the scientific literature, it's been, proposed that that we may be able to revive extinct species. And today, with advances in genomics and synthetic biology and CRISPRs, some consider that it's possible to revive extinct species. And I'll just say that it's then no coincidence that the company behind de-extinction, one of George Church's company, Colossal, just hired Joanna Damas. She just started, the postdoc who was doing those studies, into the company. And so, it's clear that, the company's strategy is to use CRISPRs. And, in our mind, in my mind, it's always been rather dubious. You might be able to produce a hairy elephant, but it's still a hairy elephant. If you really want to produce a mammal or a thylacine, you really need to have a genome, a proper type of genome reconstruction to do that. So I would argue reconstruction before reproduction. Okay, in the closing minutes, I'm just going to say that the project, the Earth Biogenome Project, has many challenges ahead of it. The most significant challenge is the collection and vouchering and storage of samples for sequencing. This is not a trivial problem, and one which we are actively engaged in both technological solutions and in ways that we can spread this to citizen scientists to broaden the participation in the project. There are problems from sample collection, there are problems in computation, there are problems in chemistries and many problems along the chain. And we are actively working to solve those problems so that we can advance from phase 1 to phase 2, because to go from phase 1, which is 9 genomes per day, to phase 2 is 125 genomes today. That's a 14-fold increase in throughput in the world. And we're going to need to have technological advancement. So it's a Sisyphean challenge to push that ball up the hill. Many challenges still ahead of us. And we have a big workshop in May to begin to plan to overcome the bottlenecks for phase 2. So it's not going to be easy, we know that, but we are clearly through the pilot phase. We have our liftoff for our moonshot in biology. We understand we have 1,000, we need 1.8 million. We have a long way to go, but we have begun our journey, and there is no turning back at this point. Okay, so I want to thank you, and I want to just take one more minute to thank the 2 postdocs, Joanna Damas and Marco Corbo, who have been involved here at the University of California, Davis with me in our chromosome evolution studies. Our chromosome evolution working group, I mentioned Jae-Boom Kim and Jian Ma, the algorithm developers for G-Scrambler, Marta Ferre, at the University of Kent, Dennis Larkin at the Royal Veterinary College in London, and then the many groups who have supplied genomes to us for these studies. And importantly, and this could not have been done without the people at the San Diego Zoo, Ollie Ryder, in particular, Cynthia Steiner and Marilis Hauk, to provide us really high quality samples for high quality DNA extraction for the long reads that we needed to do these chromosome scale assemblies, and all of my colleagues in the Earth Biogenome Project and the Vertebrate Genomes Project. And with that, Fernando, I will thank everyone for attending and close my talk this afternoon now in your time. So thanks very much.
This was fantastic. Thank you so much. If you'd be okay with that, I have one question in the chat and then 2 now. Then I will just move forward if anyone has. I don't know from Charles, can people speak here or not? Or just a chat? Well,
They cannot speak, they can only chat. They can raise their hand and I can unmute them if they need to be unmuted, if they'd like to talk.
So they can speak the questions. Okay. So I have 2 questions on the chat. I'll start with them and then we can move from there, and I'll have my own question. So the first question we have here is, when collecting samples of each species to be sequenced and annotated, is a single individual collected as a representative of the whole species, or are multiple individuals collected and sequenced to get a more comprehensive perception of a reference genome?
Yeah, great question. And sorry, I had to gloss over so many issues related to such a big project. But the answer to the question is yes. There's a single individual for the reference genome. We are not doing at this time pan-genome as being done for other species, but for all of the threatened and endangered species, we have included in the design population levels here. At least 10 individuals from each species will be resequenced to determine population level genetic diversity, such that we can understand what the levels of genetic diversity are in these threatened and endangered population. So for reference genome at the moment, it's one species with long reads plus high C. And for threatened and endangered species, like in the California Conservation Genomics Program, I just might add, we're doing, Brad, that's led by Brad Schaefer at UCLA. We're doing for that 150 individuals per species in different populations across the state of California. There's a $10 million project funded by the state of California for conservation genomics. So there are 150 reference genomes. No, I said there are 150 reference genomes, then 100 individuals per species, not 150. 150 reference genomes, 100 species in different populations across the state that can then be used for to develop conservation management plans for each of those species.
Thank you. And that question came from Tiffany Roach. Thank you, Tiffany.
Thank you, Tiffany.
The second question from Sarah Graham. To clarify, you said that the regions that are highly conserved relate to anatomical and central nervous systems, while the breakpoints are associated to sensory perception and regulation of transcription.
Yeah, that's close to what I said. What it is that the genes, yes, the genes in those conserved blocks are related to anatomical and central nervous system, and really the entire developmental plan of amniotes. Most of these blocks go all the way back to chickens. And the regions between the blocks, you're correct there, have genes that are related to adaptive functions, sensory perception, immunological functions, and many novel transcription factors appear in the regions of breakpoints. And this is really because when and segmental duplications, one of the really important charts that I didn't show that goes back to our work in 2009, is that the breakpoint regions are highly enriched for segmental duplications. And that's extremely important. As you look in the different species, you can see tremendous variation in those copy numbers of those segmentally duplicated genes in those regions, because when the chromosomes break and they repair, that's the likely mechanism by which those segmental duplications are expanding and contracting after those chromosome rearrangements. Yeah.
Thank you so much. That question came from Sarah Graham.
Thank you, Sarah.
The next question from Claire Gleason. I might have missed it, but is there a certain set of criteria that you follow to decide which species genome you sequence sooner rather than later?
Yeah, so the priority is first taxonomic family level, then at the genus level, and then at the species level. Now, within the families, we don't prioritize them, but the reality is that funding prioritizes for us. So for example, NIH just came along and said, hey, we want you to sequence all of the models associated with human aging. Okay, so those organisms get done. USDA is interested in funding anything you can eat or things that will make the things you eat sick, plants and animals. And so they fund, they have a big project called the AgPest 100 project, which is animal, mostly plant, sequencing of plant pathogens. And so it kind of goes like that. The Department of Energy is sequencing lots of plants, but they're all plants that are related to biofuels production or algae that are related to biofuels production. So in the United States, what gets sequenced is highly dependent on who's funding it and what the mission of that particular agency is. Now we're trying to break away from that, and we've been working on this in legislation in Washington to create an interagency working group of all the agencies so that we can get away from simply the mission-oriented thing, because eventually we're going to be sequencing a lot of the same families and a lot of the same within the same genus as well. And we want to keep this broad phylogenomic approach to get a representative, because that's the backbone of the tree of life, that we can hang everything on. It's going to be very important to try and stick to that plan as much as we can, but that's going to take cooperation among all the agencies, okay? And we're starting that now in the CHIPS and Science Bill, and the last executive order by President Biden is there's language that calls for the development of a national strategy for biodiversity genomic sequencing. And, you know, we understand that there are important missions of these agencies, but we hope that they can come together and cooperate and collaborate on making sure that we don't leave any parts of the tree out. Okay.
Thank you.
Thanks for that question. Right.
And you absolutely read my mind about my question, which was going to be about how the funding agencies are responding to the prospect. That's fantastic.
Yeah.
I have another question from Igor Sharakov. I hope I did not mispronounce her name. You mentioned three-dimensional genome organization. Have you studied its evolution or its role in the evolution of gene order?
We certainly have, and we published a paper before this paper just came out on the reconstruction. There's a paper by another postdoc from my group named Marco Corbo, also published in PNAS, where we looked at the evolution of three-dimensional structure using high-C data. And we use carnivores as a model. These are not easy studies to do, but we use the carnivores, 3 different families of carnivores. We use the bears and cats and the dogs. And we compared three-dimensional structure of their chromosomes and their chromosome segments. And we found, somewhat to our surprise, that there was some very deep conservation of three-dimensional structure over more than 50 million years of carnivore evolution. The closer that you got, and if you looked at the felids and the canids, there was even greater conservation. And so not only is the order important, I think, Igor, this gets to your question. It's not just that the order is important, but these syntenic blocks are maintained as in a consistent three-dimensional structure over long periods of time, which must also be important for the regulation of the activity of the genes within those regions. And when you go across the blocks, you see a lot of fuzziness. You see a lot of compartment switching. You see A's to B's and in the inability to clearly define the three-dimensional structure or the compartments within the blocks. And we propose in that paper is that's a very interesting way of getting fine-tuned regulation that may be lineage specific after a rearrangement, is by changing the way the genes, not just changing the enhancers, of course, and the promoter, but just changing the three-dimensional architecture could also affect the activity of the genes within those blocks. So thanks for the question. And Fernando can probably get you the paper, or I can if you send me an email.
I read
Corbo,
that.
C-O-R-B-O. Yeah.
And if I'm not wrong, Igor's in my genomics class.
Oh, great. Okay.
Does anyone have any other question? Either type here or unmute yourself. I don't see any activities here on the chat. Well, if there here's another one. There's just a thank you for your fascinating talk.
I appreciate that. Thanks very much.
Yeah. So I guess we're closing this session. I cannot express how thankful I am for you to spend this time. And thank you all for joining. And if you're okay, Harris, just stick. I'll send you a note with another link for us to chat with my kids here, if that's fine with you.
Absolutely. Anybody can stay. I would say anybody who wants to stay for a discussion, If that's okay with you, Fernando, we can do that.
Yeah. But yeah, I'll send you another link.
Yeah, I want to, another link. Okay. I want to
Yeah.
thank everybody because this is a really difficult format. I've given, I don't know, hundreds of lectures online and teaching online. I don't really like it very well, but I want to thank everybody for, you know, trying to pay attention and stay with me in this kind of presentation. I know it's not easy. So thanks very much.
No, this was fantastic. So I'll forward you another link. And
Okay.
a couple of people asked for the papers. I'll take care of sharing all the papers with them myself. Okay. Thank you
Yes.
all so much. Thanks, Charles. And thank you, sir. I'll talk to you in a second.
Thank you all. Bye.
Bye-bye.